Good evening. Greetings to one and all gathered here. It gives me an immense pleasure to welcome you all to the Biotalk series version 2.5. In our midst, we have an eminent person, Dr. S. Anbaragi, who is an assistant professor in the Department of Environmental Science at Central University of Kerala. And she is going to speak on the topic foodborne illness. Now, I call upon our HOD, Dr. Sri Kumar, to inaugurate the event.
Good evening, my dear students and staff members. Hope everything is going on safe. I'm delighted to see you all again in this virtual BioTalk lecture series, version 2.5. As a part of the inauguration, let me share some of the credits of our college and the department. St. Joseph College of Engineering is one of the top colleges in India with 160th position as per the current NIRF ranking. It has got 27 years of high reputation in educational society with NBA and NAC accreditation. It has got 10 UG programs and 7 PG programs with an affiliation from Anna University, Chennai. Recently, we have got autonomous status also from UGC, which will be implemented from forthcoming academic year. Our Department of Biotechnology is started in the year 2002 and having BTech and MTech biotechnology programs in it. Since inception, we have bagged 139 university ranks and 10 golds and 7 silver medals in it. And consistently, we maintain 100% pass percentage every year. The department has got sophisticated laboratory facilities and fetched research center recognition from Anna University to conduct PhD program also. We have more than 1.2 crores of government funding for conducting research. As we all know, the current pandemic and lockdown and online mode of teaching learning procedures imparts a psychological burden on students and academicians because all the time we were mentally and physically more active than others. In my view, high priority should be given to our biotech student community, and we should keep our students to be abreast with the various avenues of research. The reason is that any innovative giant leap in the biotech research could be a salvation to the entire world from this current situation. From this, our department has learned to go for a procedure of periodically conducting BioTalk lectures by bringing eminent personalities through webinar and sharing their research views to the entire biotech students, teachers, and research personnel of our college. In this order, today we have fifth BioTalk lecture in the topic of foodborne diseases, which is a most relevant and the need of our topic as far as the current situation is concerned. As per WHO report, the surprising fact is that almost 1 in 10 people in the world fall ill due to the foodborne diseases. The World Bank also estimates that 110 US billion dollars is the loss per year due to the medical expenses resulting from an unsafe food consumption. If we understand the nature of foodborne diseases, we can develop a safe food handling method, food supply chain, and food preservation methods, etc. So that we can surely decrease this illness, mortality rate, and of course the expenditure towards medication. In order to give more authentic research-based explanation about foodborne diseases, today we have Dr. Anbaragi, who is an assistant professor of the Department of Environmental Science from Central University of Kerala. On behalf of the patrons of the college, Dr. B. Babu Manohar, Chairman, Madam Jessie Priya, Managing Director, and Mr. Sasi Shekhar, Director, Dr. V. S. Rao, Principal, and Dr. Parvadavardhini, Dean. I welcome the speaker, Dr. Anbaragi, and all the participants and staff members to this BioTalk lecture series. I also convey my warm felicitation to Dr. V. Renuka, Professor, and Ms. G. Kalavati, Assistant Professor, for organizing this webinar. All the best to you all. Now I ask the student coordinator, Mr. Alfred Francis, to give a brief introduction about the invited speaker. Over to Mr. Alfred.
Thank you, sir. Dr. S. Anbaragi has done BSc Botany from University of Madras and also completed a MS in Ecology from Salim Ali School of Ecology, Pondicherry. And also she has done a PG Diploma in Marine Environment, Pollution and Management in Nanobiotechnology from Annamalai University, Tamil Nadu, and also from the Center of Life Sciences, Bangalore. She has also done a doctorate of philosophy on ecology from National Environmental Engineering Research Institute, Chennai. She has also done postdoctoral research on environmental sciences from Bharatiyar University, Center of Life Sciences, Coimbatore, Tamil Nadu. She has started a career as a biology student in Rosalie Matriculation Higher Secondary School, Chennai, and joined as a project assistant at National Environmental Engineering Research Institute, Chennai, and was later promoted as a senior research fellow in the same institution. Later, she joined as a principal investigator in the Bharatiyar University, Coimbatore, in the DRDO BU Center. Currently, she is an assistant professor in the Department of Environmental Sciences at the Central University of Kerala. She has involved herself in many projects such as concentration and detection of human enteric virus in marine environment sponsored by the Department of Biotechnology, Government of India, and also the project on the study of water quality surveillance in Tamil Nadu sponsored by the Hindustan Level Limited Research Center, Mumbai, and many more. Dr. S. Anbaragi has published many more books of chapters on supercritical water gasification, a pilot study on the hotspot regions of marine environmental pollution along the coastal regions of Chennai. She has also published many journals on prevalence and factors associated with multi-drug resistant tuberculosis in South India, and also the detection of typhoid fever by diatom-based optical biosensor and groundwater quality assessment among the selected blocks of Wayanad district and many more. It is an immense pleasure in welcoming you, ma'am, and I request you to take over the session from here.
Thank you so much. Professor Renu, can I proceed?
Yes, ma'am.
Yeah. I would like to thank Professor Srikumar sir for the warm introduction and Mr. Alfred, the students coordinator. Thank you so much for the introduction, Mr. Alfred. And can I share my screen now?
Yes, ma'am. Sure, ma'am.
Yeah. Is it visible?
Yes, ma'am. It is visible, ma'am.
Okay, thank you. Yeah, as mentioned myself, Dr. Anbaragi, have done some couple of projects related with foodborne pathogens. Especially I have worked with Listeria monocytogenes. It was sponsored by the Department of Science and Technology, where we tried to inactivate the Listeria monocytogenes before it reaches the consumer. So one aspect we have done with that. Another project I have done with the same DST, where we tried to inactivate any foodborne pathogen, especially bacterial pathogens, using UV LEDs, light emitting diode. We try to incorporate in household refrigerators, where we try to completely inactivate and increase the shelf life of the produce. So based on that aspect, I would like to give a talk here on a lecture, you can say, on foodborne illness. Before starting my lecture, there is a quote I would like to mention here. When you talk about foodborne pathogens, the food that you eat, they can either be the safest, the most powerful form of medicine, or the either end, it can be a slowest form of poison, isn't it? So as mentioned by the professor and head, that 1 in like out of 10 across the globe, they are suffering out of foodborne illness. Yes, and several millions of economy is lost because of that. It's a very valid truth, actually, that is prevalent. Thank you for the information, Dr. Srikumar sir. And so moving into the lecture here, when you talk about foodborne illness, we are talking right now, or maybe from the 19th century onwards. It's not like that. The foodborne illness started way back in the Hippocrates period, that is in the ancient Greek period itself. It started actually. Then during the 19th century, there were several incidents of contamination in food because of milk, because of some other food, because of water. It all started. And then slowly the sanitary revolution started. So because of the sanitary revolution, there was an awareness that water must be treated, sewage must be treated, and washing practice must be followed. And pasteurization of milk came. We all know because of brucellosis, there is a chance of contamination in raw milk. So after pasteurization, all this awareness on foodborne illness once again start. And again, we all know refrigerators came in 1913 onwards. Then this kind of awareness about these kind of pathogens came into the sight of the people. Okay, right. So what is actually foodborne illness? So when we say foodborne illness, any infection, an infectious disease which normally spread by consuming a contaminated food or a contaminated beverage. So there are nearly 250 known foodborne diseases are there. These foodborne illnesses are caused by mostly by bacteria, sometimes by virus, sometimes by parasites, and also by the toxins produced by these bacteria. So we can say that it's an infection because of these above mentioned reasons. So when you talk about the etiology of this foodborne disease, and as I mentioned, it is because of exposure to these pathogenic microorganisms. Maybe there will be an acute effect on the human health and sometimes chronic also. And as mentioned above, it's not only because of bacteria, virus, we come in the order, virus, bacteria, parasites. It can also be because of chemical substances. We know the residues of pesticides, lots of veterinary drugs we are using, number of additives, adulterants are there in our food, lots of toxins are there, biotoxins, mycotoxins. They are into our food intentionally or unintentionally. These are also the reasons of foodborne diseases. So the problem starts when you consume them. Any contaminated food, it is going to be detrimental to our health. Right. So this data was already mentioned by the professor and head, that 1 in 10 fall ill and lots of death. And that too because of diarrhea, which is caused because of acute gastroenteritis. Several lakhs of deaths are occurring. Especially the deaths are very common below the children of age 5. They are prone to that. And also in case of adults, those who are immunocompromised, that we will see in the forthcoming slides. So the WHO estimates that 1 in 3 people worldwide, they suffer from this foodborne disease every year. And 1 in 10 is a sufferer every day, right? Okay. So what is the reason for this food contamination? There are several reasons, right? See, food now here in this global situation, it's very difficult to point out where the contamination has started. From farm to fork, okay? From somewhere across the country, in some part, somewhere the food is produced, right? It is being like marketing, it reaches somewhere else. See, we eat different varieties of food. We sometimes when there is an infection for us, we don't even know which food has exactly causes like some kind of infection to us. isn't it? So because somewhere it is produced, somewhere it is processed, somewhere it is marketed, somewhere it reaches some part of the world, it reaches a consumer. And several ingredients we are using, we don't know exactly to point out. Many times in developing countries, these are underreported, sometimes not reported. Because of this kind of pandemic situations now, this has created lots and lots of awareness among people that what to eat, what not to eat, how to eat, where to eat, isn't it? Now, most of us are very familiar with the word hand washing, sanitizing, disinfection. We talk asymptomatic area, lots and terms, quarantine are very familiar now, isn't it? But earlier, there is no such kind of awareness among people. So how this food contamination occurs? When you take maybe from the raw material, maybe fresh fruit or uncooked food, ready to eat food you may be eating, that food would have been grown in a form where fertilizer has been used as from animal manure. Maybe those producers would have been washed with a contaminated water, right? Maybe while transporting it, it would have been handled by a person who have been infected, handling by an infected person. Maybe the produce would have been good, by when packaging it would have got spoiled, isn't it? So there may be animals that would have acted as carriers for these pathogens. There can be several reasons, right? So some foods which comes from the store, they may contain potentially harmful microorganisms. By not washing your hands before cooking, not storing your food in the particular temperature which is prescribed, that we will see in the forthcoming slides. Not thoroughly cooking your food, that is a contaminated one. Not properly defrosting, thawing your foods, not cooling your food which is supposed to be quickly cooled, or not cleaning your surfaces before starting the preparations. There may be several reasons for this contamination. So this is a very important slide. We should know how this transmission occurs. Okay, so you can see a F diagram. The F diagram says there are 5 F's, the reasons how a contaminated food transfers. Normally, the foodborne illness occurs by fecal oral route. We can say oral fecal route, orophycal route. Okay, any food which is contaminated by a feces, that can enter your food, that can enter your fingers, that can enter your fluids, that can be in through the flies, it can reach your food, from the field it can reach your food. That is how a food gets contaminated. How these feces or these pathogens, through these modes, they enter your food, and that is how you get infected. So do all get infected? No, not necessarily. It all depends, as you know. Now this time makes everybody understand the kind of infection to whom it is prone to. So vulnerable people, you can see the picture, a pregnant woman, very young children, maybe there are 2 categories you can say, under 5, under 10, and then very old people whose immune system is compromised. Maybe you can be a patient of HIV or a kidney failure patient, you can be diabetic, or you can be organ transplanted individual. So those persons whose system of immunity is weakened, they are all vulnerable to this foodborne infections, right? Okay, so let us know about these foodborne pathogens here. There are lots of, I told you there are lots of categories like bacteria, virus, parasites. Of course, the most common are the bacteria. Here, they are the most common one in causing foodborne illness. And bacteria, we say, we all know that they exist in lots of shapes, types. They have several properties. Some are spore forming, non-spore forming. Some of the bacteria are highly heat resistant, and some can even produce heat resistant toxins like Clostridium botulinum, right? And most of these bacteria, they are mesophilic. They can grow in their optimal growth temperature is our body temperature. 20 degrees to 45 degrees is the range. Of course, when they enter our system, they are very convenient in multiplying, right? And some of them on the either extremes in case of foodborne pathogens, they are even psychotrophs. They can even grow in a temperature less than 10 degrees. So storing those foods which are contaminated also is a question mark here. When there is a food contaminated with Yersinia or a food contaminated with Listeria, when you are going to keep them in a refrigerated conditions, there is no guarantee that the food is safe for us to consume without heating it once again. Okay, right. So let us know some of the prominent foodborne bacteria here. Bacillus cereus is the one. We all know they are gram-positive bacteria. They are motile. They have spores, and they can be seen throughout the environment. It can be a soil environment, fresh water, marine water, and they can be seen everywhere, and they can grow easily elsewhere. And whenever the moisture is there, whenever protein is there in your food, they can grow very safely and they can cause lots of infections. And the infection, you say like there is an incubation period. See, whenever you eat a contaminated food, the moment you eat, the moment the symptoms are getting exposed. This we call it as an incubation period. The onset of the symptoms, the exposure to the food. That period we call it as an incubation period, which is less than 24 hours. So between 8 to 16 hours is their incubation period, and they will show symptoms like abdominal pain will be there, nausea, you feel like vomiting, diarrhea. Soon after 8 to 16 hours of eating a contaminated food with bacillus. Then another prominent bacteria is Campylobacter. Campylobacter is again the symptoms will last for 2 to 5 days a year. Sometimes the severity ranges like none at all, sometimes even to death. So all these kinds of diseases are prevalent whenever you are eating raw food or what do you say, like undercooked meat items especially. And this kind of food eating practice is less common in our country because Indian cuisine style of cooking is something different, where we cook it in like maximum temperature. The internal temperature is very, very important in cooking. So as far as whenever, if at all you get that infection, maybe you would have eaten a salad or a fruit, which would have been handled by an infected person in the hotel or the restaurant wherever you eat. Or else the cooking style is completely safe in our Indian environment. Okay, so maybe because of a contaminated water, unpasteurized milk, and because sometimes when we take juices from the juice shops, have you ever witnessed they take the milk, cut it, and directly they will pour the milk, right? So they never boil the milk, and they prepare a boiled and a cooled milk will never be used for rose milk or whatever you take, isn't it? So if at all the milk is a pasteurized milk, is good, right? Okay, so contamination can happen through unpasteurized milk, contaminated meat, maybe the reason for Campylobacter. Then we have E. coli. You can have a doubt here. E. coli, we all know that it is a natural flora, and it is found in the digestive tract of cattle and other animals also. Of course, some of them are very, very dangerous. We know that, right? So there are several types in this. We can say like there are 6 groups in E. coli which can act as pathogens. You would have heard about E. coli O157H7 strain. E. coli O157H7. They produce Shiga toxin, which is very, very dangerous. And as I mentioned, there are 6 groups in E. coli. Enteropathogenic E. coli, we say. Enterohemorrhagic E. coli. That is, they produce the Shiga toxin. And enterotoxigenic, enteroaggregative, we say. Enteroinvasive E. coli. And lots of attaching and effacing E. coli. There are several types of E. coli which are very, very dangerous also. Okay. Then comes Shigella. In case of Shigella, this is also like they produces a toxin, and this Shigella is also belongs to the family Enterobacteriaceae. There are several serotypes in that. They are found wherever the hygienic conditions are very poor. You can see this particular bacteria. The primary route of transmission is person to person, and whenever there is an ingestion of fecally contaminated food and water, you are prone to Shigella. Okay, the incubation period is maybe after 1.5 days, you may have this infection. By that time, you may not even know. You may be thinking the previous food you have taken, but the reason would have been 2 days before you would have eaten something else, which would have caused this kind of infection to you. And here you will have again several symptoms like fever, you will feel very weak, and there will be cramps, dysentery. The only problem in our country is normally whenever we have a diarrhea, we never go get our stool tested, isn't it? Either we take rest or the diagnosis is never done, and we never look into the root cause. If at all also, whenever we give our stools for culturing, it is not of bacteria. There is no like next level of taking the diagnosis. Is it because of any other parasite? Is it because of a virus? That kind of diagnostic next level we never go. We go to a doctor, maximum we will get rehydrated by IV, we get intravenous fluid, or we get a dose of antibiotic. By the time we get like ourselves self-cure. So we don't go to the next level of diagnosing or reporting or giving the data to the government, right? Maybe here after this kind of COVID, we may be having lots of awareness, isn't it? Next prominent foodborne bacteria, where it happens among the formula, the baby foods, Chronobacter sakazakii, we say, and this is also a gram-negative bacilli, and this was first isolated in a powdered formula. It can cause severe neural problems with neonates, right? And they can grow in 30 degrees very well. Even in our own 98.6 degrees Celsius body temperatures, they can easily flourish. And they not only with the neonates, they are capable of causing infections, as I mentioned, among the elderly people and immunocompromised adults. And it has been widely across the globe, it has been detected in the infant formula. So a very good manufacturing practice is a must. In canned foods and formula foods also, don't think there are no chances of infection. One has to be very careful in preparing even the infant formula in hot water with sterile condition. How you are going to prepare and giving to your child is very important. Then, as I mentioned, Listeria monocytogenes. This is a psychotrophic bacteria. See, this bacteria is very, very dangerous. If some infection happens to a pregnant woman, all of a sudden it can cause abortion, miscarriage. Sometimes it is capable of causing meningitis. It can inflame your brain. So lots of dangerous effects are happening because of Listeria. Maybe all these data are available in developed countries. In developing country, when such incidents happens, we never go test what is the reason behind such an abortion or some reason, isn't it? So we think some other reason, we never correlate with the food we have eaten a few days before or something. We never try to correlate them. So Listeria monocytogenes, if it is present in a food and the food is contaminated, even if you are going to refrigerate your food item, they can grow. You cannot arrest its growth like it's another mesophilic bacteria. Okay, right. Then the very famous typhoid causing Salmonella. Salmonella causes typhoid, most of us know. Through poultry, it reaches us. And the symptoms, like it all depends. If it is a typhoid, the period of suffering period, the infection period is very long. It can cause lots of other kind of damage to your organs can happen because of such infection. All you have to know that whether all these foodborne pathogens can be prevented or not. Yes, definitely these infections can be prevented by simple steps like thorough washing of your hands and keeping your food out of the danger zone. Danger zone, we mentioned, a danger zone is nothing but try to keep your food between 4 degree and 60 degree. So if you want to store something below 4 degree, keep it in freezer. And don't ever leave your food like for 2 hours in your dining table or outside. After cooking, beyond 2 hours, never try to keep your food outside. Okay, if at all you want to eat them, store them in the right temperature of fridge or freezer, or try to reheat it and make it sure that the internal temperature rises to 60 degree, and never try to reheat your food more than once. Like once while cooking it reaches and again reheating. So beyond that, 2-3 times, never try to reheat your food and eat. That is also not good if some toxin has already been there, right? So never leave your food, cooked food for more than 2 hours in room temperature, because we all know that like they can regenerate, like they can multiply. The generation time is 20 to 30 minutes in case of bacteria. So they can efficiently multiply with the help of the moisture in your food and the nutrient in your food, right? Then we have another bacteria, Staphylococcus aureus, and this also like improper handling of the food. Most likely they are present in poultry, that is meat, eggs, poultry, and chicken. Yes, of course, these can also be prevented by proper hand washing practice and thorough cooking of your food at perfect temperature, internal temperature of the food. The food temperature, in the sense, every part of the food should attain that temperature. Then comes Yersinia enterocolitica. This is also a psychotrophic bacteria which can grow at below 10 degrees Celsius. And these are gram-negative and non-spore forming. The one good aspect here, they can grow in both aerobic and anaerobic conditions, right? So they are widely distributed throughout the environment. In sewage, you can see them, in seafoods, raw milk, you can see them elsewhere, and importantly in pigs, right? So it's very dangerous. There is no guarantee if you are going to refrigerate the food which is contaminated. They can easily grow in that temperature, right? So you can say that Yersinia can grow between 0 to 45 degree, and obviously the optimum temperature is 25 to 30 degree for them. They can grow very well in alkaline conditions also. This is another dangerous aspect. And how to overcome? There is lots of bacteria. We were talking like they are causing foodborne illness. They can cause diarrhea. They can infect individuals, young ones, adults. Yes. What is really allowing this bacteria to grow this much comfortably? You have to know this theory in food science, fat Tom. Remember this word, fat Tom. These 6 factors are allowing your bacteria to grow comfortably in your food. The food which is present is the only source for them to grow, right? So the nutritive food, the moisture in your food, and the proteins in your food will be favoring their growth, right? Then what bacteria need to grow? You can say acidity, right? In an acidic environment, obviously they cannot grow. In near neutral conditions, they are very comfortable in growing. That is why like pickles and all will be very acidic. They cannot grow in that. And then another condition which is favorable for the bacteria to grow. See, I told you that bacteria, they can grow the best in foods that contain very little acid conditions only. So we know the pH ranges of 0 to 14, and 0 is highly acidic and the value 14 is highly alkaline. We know that pH 7 is neutral, and bacteria always grow the best in neutral condition and slightly acidic condition also. right? Then comes time. How long you're exposing your food, how long it is being allowed to left. As I mentioned that in that particular temperature of room temperature, if you're going to allow the food, contaminated food, remain as such, comfortably they will multiply and grow. So the exposure temperature time is also very, very important. As I mentioned that temperature, bacteria grow quickly between the temperature of 5 degrees Celsius and even to 57 degrees Celsius. So this range is, we call it as a danger zone. Danger zone is 40 degree Fahrenheit to 140 Fahrenheit. When you say in Celsius, it is 5 degree to 57 degrees Celsius. So we call in food science it as a danger zone temperature. Then bacteria can grow very rapidly also in this particular temperature, and we know the availability of oxygen. Some bacteria are also anaerobic. When talking about the moisture, water content, more the water content, very safe for the bacteria to thrive. All the bacteria need some amount of water content in their cells to grow, and more the moisture. That's why always we go for we salt salty foods, dry. We make we dry the foods and keep them so that no bacteria will allow. So we use ice salt for storing foods, right? Dry fishes, we use more salt, dry them in sunlight, and so that bacterial contamination will not be there in that particular food, which has to be stored for longer period of time, right? So fat term, you need to remember food, acidic condition, the time, the temperature, oxygen level, and the moisture content are the factors which affects the bacterial growth. So moving on to foodborne viruses, there are several foodborne viruses. The prominent ones here are norovirus and hepatitis A virus. In case of hepatitis, there are A, E, B. Hepatitis B is bloodborne. Hepatitis A and E are waterborne. We have worked with that, and we have publications also that. And I have worked with norovirus, with the RT-PCR detection of norovirus in Tamil Nadu, and throughout India we have collected samples, and that was my PhD when I was in NEERI CSIR Taramani campus. So where we try to isolate noroviruses for the first time in India, and it was a strain from Japan. So here the foodborne viruses, we know the difference here is bacteria multiply. Food viruses, they don't multiply. They keep inert. Whenever there is an available host, they start multiplying after entering their system. We all know that, isn't it? So foodborne viruses, when you talk about, there are nearly 100 types of enteric viruses, and the most common, as I mentioned, foodborne virus are hepatitis A and norovirus. And these viruses are transmitted via food, and especially the clams, molluscans, bivalves, oysters, which we eat. There are two types of fish we all know, fin fish and shellfish. Shellfishes, normally they are all filter feeders. They gulp liters and liters of water every day, and through that they collect their food. And sometimes they try to exit the foreign particle. When the accumulation is more, they cannot eliminate them, right? We call this as depuration. That is not possible, so they get accumulated inside the body. Most of these bivalves are eaten raw or undercooked in foreign countries. They eat them as such. They love to eat them, and most of the outbreaks are common in those such countries where there is a practice of eating lots and lots of oysters, bivalves raw or partially, slightly cooked. They like a salad, they try to eat them. As a ready to eat, they eat them, right? That kind of practice is not here. Whenever there is an accumulation in that inside the system, you are prone to a foodborne viral infection, right? So it is all depending, for example, how this clams gets this virus. Again, if that clams are growing in a contaminated water, wherever there is a fecal contamination, wherever there is a fecal sewage discharge, when you are going to harvest those shellfish from that zone, obviously, since they are filter feeders, they will be having these viruses inside their system. Okay. So normally shellfish are usually eaten with their digestive tracts in place. We don't remove them, and sometimes lightly they cook it and raw they try to eat. So again, thermal inactivation is very important. How far, in what temperature, how long you are cooking matters a lot, right? Okay. So when you go to norovirus, earlier the name was Norwalk virus. Norovirus, we call it as a winter vomiting disease. But of course, it happens even in summer also. These viruses, they cause gastroenteritis among elders as well as children. There is another virus you may be familiar, rotavirus, isn't it? So rotavirus, very common among children under age 5. Here, this norovirus is also like human feces infected. If you are going to eat them, definitely you will be getting this foodborne illness because of this virus. So how this actually happens, not only because of a shellfish which is growing in contaminated zones, it is also if a staff is having a diarrhea or vomiting, if he is having noroviral infection, if he is going to handle the food, obviously it will also get from one person to another person, right? So norovirus is commonly linked with ready to eat food also. In contaminated water. So norovirus is often transmitted, like transferred to food by an infected food handler, or with if they are if they are using a contaminated gloves or a finger cuts, if that they are all contaminated with feces, then the another person will also become sick. So it is very contagious, and people become contagious within a few hours after eating them. The virus is often in a person's feces for days. Like after the symptoms have ended also, it will remain in your feces, right? After like the diarrhea is over also, few days you will be shedding that virus in your feces. So we all know that 1 gram of feces will have several millions of viral particles, isn't it? Maybe if that person is in a restaurant, like he is going to a washroom, not thoroughly washing his hands, then the handles will get contaminated, isn't it? The doors. Nowadays we are all aware how to open a door, how to use a lift, how to use the buttons. Everything we are familiar after an infection, which is prevalent for a longer period of time. Most of them have got the knowledge, like how to use them, right? So hepatitis A virus, which causes inflammation of your liver. Here also the symptoms like fever, you will become tired. Obviously jaundice will be there, and this symptoms will be for like few days, like 30 days after contact you will be getting. See, then just think about it. How you will be tracing, you will never be analyzing what food, where you have eaten, what has brought this kind of infection to you, right? So 30 days of contact after contact you will be getting the symptoms. So this particular hepatitis A virus will stay in your bloodstream for about 6 months. And hepatitis A virus will enter through the mouth, they will multiply in your body, and it will be spread by not proper clean hands, especially after going to the restrooms, bathrooms, we can say. So food, they are found in any foods touched by infected individuals. They can easily spread by flies also. So shellfish which are grown in contaminated water, definitely they may be carrying hepatitis A virus. Of course, you can prevent them by a simple practice of thoroughly washing your hands, thoroughly cooking your food in the perfect zone. Then, apart from bacteria and virus, it is also caused by parasites also. There are lots of parasites. Like protozoans, which is also capable of causing foodborne illness. We have publications with Cryptosporidium and Giardia. We have worked with that when we were working with the Pureit. We all know the Pureit water, right? In that, like what do we have to say? In the in collaboration with Hindustan Lever, where we try to validate a Pureit for virus. We try to validate it. We can't grow all these viruses, right? We try to work with surrogates. We try to work with bacteriophages. We can't grow all the enteric viruses, right? Cell lines are required. All viruses don't have cell lines. We can't grow them. So in order to validate the purate, we worked with bacteriophages, and that time we had the opportunity to work with the Cryptosporidium and Giardia using fluorescent microscopes. We try to isolate them. Okay, so foodborne parasites, we all know parasites are one-cell microorganisms and without a rigid cell wall, and they have an organized nucleus. Of course, they are larger than bacteria, and like viruses, they do not multiply in the food but only in the host. Okay, like they don't multiply in the foods, inside the host they multiply, and the one problem here is the cyst, right? So the transmissible form of this organism we call through cyst. Through the cyst it enters. Then parasites, they get their nourishment and protection from their host, and they may be transmitted from animals to humans, human to human, human to animals, and very common is this one in country like United States of America. Here particularly, most of them would have faced this infection, this particular foodborne parasite, Toxoplasma gondii. This Toxoplasma gondii is it's an intracellular protozoa, and it is one of the 3 leading causes of death from a foodborne disease. Okay, so you may ask, what are the other 2? Salmonella and Listeria. Okay, right. So who are at greatest risk to this toxoplasmosis, or again pregnant women, immunocompromised individuals, HIV positive patients, right? So the source of this particular protozoan toxoplasma is infected cat. They shed them in their feces. Soil, they will reach the soil, undercooked meat, and there are vectors for them, cockroaches, okay, and flies. And if you see the clinical symptoms, humans are asymptomatic, okay, they don't show any symptoms, and if at all, fever will be there, headache, and there will be swollen lymph nodes, right? So what you have to do is you have to prevent yourself from this infection. Whenever you do gardening, you have to wear gloves, because as I said, it will be there in the cat litter. And here, having pets as cats is very common in those countries. Here we don't keep them like of course cats are there, but if at all, you have to be little careful. That is it. Thoroughly washing the fruits and vegetables before eating is very, very important. And you have to see to that not 60 degrees is a guarantee. You have to increase the temperature even to 73 degrees Celsius or 80 degrees Celsius. You have to cook. If at all, if you are suspecting your food with toxoplasma. So it is always better to thoroughly cook your food. So this kind of practice you would have witnessed in our like elders, like the cooking style of fish curry and all in our way of cooking. They will allow the fish curry to boil for a longer time. They allow the gravy alone to boil for a longer time. Then they will add the fishes into that, and then they don't cook it for a longer period of time, 5, 10, maximum 5 minutes. They will close it and switch off, turn off the gas, isn't it? So this shows the way of cooking to arrest this kind of pathogens, right? So inactivation of the pathogens are possible by following a good cooking practice. Then comes Cyclospora catanensis. These are also protozoan parasite, and they are also they shed as oocyst, and they are also again they can cause prolonged illness to you, maybe 6 weeks, even longer, more than 1.5 months you have to suffer with this illness if you get an infection of Cyclospora. And lots of common symptoms, you won't have a bloody diarrhea, non-bloody diarrhea will be there, and vomiting. See, just think about the condition. If you're having this for more than 1.5 months, it's very discomfort to you, isn't it? Right. Then Trichinella spiralis. This is also another parasitic round worms. This causes trichinosis infection among human. They not only infect us. Most carnivorous mammals are also prone to this particular Trichinella, and again they are all capable of causing lots of infection to you. And this kind of Trichinella spiralis are viable for up to 10 years. That is the beauty of this particular one, and the incubation period is 3 to 14 days, and there won't be any specific gastroenteritis as your symptom also. So they're all very, you cannot, through thermal systems methods, you cannot inactivate this kind of round worms. That is also there. And especially people have the practice of eating pork. They are prone to Trichinella spiralis infection. And it is always better, as I mentioned, when you are eating pork, by cooking and eating on your own, try to cook it to a maximum temperature of 70 to 80 degrees Celsius for some period of time, so that there will be a complete cooking and you are safe to eat it. And as far as possible, try to avoid eating from outside, and because unless you are not sure of the cooking methods, it is better to avoid, because we don't know what is the infection they are going to bring to us. That is the way of protecting oneself from trichinosis. Okay. So not only virus, bacteria, and parasites, here comes yeast, mold, and mushrooms. We are all very familiar, isn't it? They produce some kind of the toxins which is not good to you, and sometimes it is also fatal in case of mushrooms. And we are also familiar with this yeast and other molds in our food, and these are also capable of causing, when you talk about this, what is the kind of infection they are going to bring to you? Sometimes you will end up, whenever you have to be very careful in eating what kind of mushroom you are eating, right? So some kind of mushrooms, they will have psilocybin or psilocin. They are agents we call as agonists. This agonists are capable of causing illness to you, and you have to be very careful whenever you eat. What mushrooms you are eating is very important to know, right? So then you must be familiar with another biological toxins. See, we were talking about microbes. Now you have to know about the toxins also. Normally, fish will be, when they catch the fish, if the fish dies, and before, if it takes a longer period of time to, once the fish dies, then if it takes a long time to put them in the ice to preserve them, in that period, there is a chance of histamine. Like this histidine, like the bacteria which are natively present in them, what they will do, they will start multiplying using the histidine, and they will produce histamine, which is a poison. And there is no guarantee that cooking is going to protect you from this histamine. Okay, so there is an heat shock because of this shock in the body. Like the fish, what do you have to do then? Always try to buy fish which is preserved thoroughly, properly. Those fish you try to procure from, what do you have to say, a certified, from a certified place. Don't compromise with the rate of the fish. So the fish, you have to be, after catching it, has to be quickly preserved in the right temperature. That is very, very important. If at all there is a chance of ciguatera, right? Like scompro, we need not have to get into the detail of it. And all you have to know is there are toxins in the fish also. Beyond the microbes, toxins in the fish. If, when you eat seafood, you have to be little careful. You should have the knowledge in what temperature the fish has been preserved till it reaches the consumer end. whether it has been in the refrigerated condition, whether it is in the freezer or in that frozen condition, and how it is being cooked. Never compromise with that, or else you will end up in an infection which is very, very dangerous to your health. Then apart from that, we are all familiar with chemical contaminants in our food, right? Lots of chemical contaminants are there, intentionally or unintentionally or naturally occurring. See, we are using lots of, like nowadays we have this practice of using non-stick pans, isn't it? Non-stick tawas, for even for the quick cooking practice and easy way of frying and cleaning, we always prefer them instead of iron pans. Maintenance is very difficult, isn't it? The one part here is these non-stick tawas are all made up of perfluoroalkyl substances, right? See, a very easy example of this perfluoroalkyl substances I can tell you. We are buying pizza in a box, isn't it? Have you ever seen that box will be very shining and it will be like non-sticky? Nothing will get stick into that, isn't it? Have you ever observed that? These are all perfluoroalkyl substances. Exposure of your food to that, even in picograms, if you are getting exposed over a period of time, that accumulation is carcinogenic, right? That is a different aspect. We are talking about foodborne illness in another aspect, but of course, all the perfluoroalkyl substances are very dangerous, even in pico level, even in picogram of exposure for a longer period of time. It's very, very dangerous. The non-stick tawas which we use, all. So apart from this PFAS, we are working in a PFAS project also, and apart from this PFAS also, we are all be knowing, like we are using pesticides, we are using lots and lots of like chemicals we are using. All these like are getting exposed, and lots of like heavy metals, the foods are getting exposed to heavy metals. All these are there in your, so the chemical contaminants can happen through the equipment, the utensils, whatever we use for our food. So one has to have a thorough knowledge, how to use, what for to use. Okay. So apart from that, our food also get contaminated by these nails, pins, air, all the dust, flies. All these are also like these are all the common objects which will be there. Maybe it is all because of wrong practice of handling your food. They may enter and they can cause infections to you, right? So these are all some. There are also like deliberate contamination of food is there, where we can call that as food terrorism. Groups who may attempt to contaminate the food. So we here use a term in deliberate food contamination as alert. Here the acronym alert is used, that will help you to identify the points in your operation where the food is at risk. Okay. So what is this alert? Make sure products received are from the safe source. Okay. Look, that is monitor the security of the products in the facility. Who is handling is very important. How you are keeping the records of the food is very important. Develop a plan for responding if there is any suspicious activity. So we call this as alert. Assure, look, employees, report, and threat. So this is the best way to protect food, is to make it as difficult as possible for someone to tamper the food. So defense is very important. Food defense program is very important to keep our food out of risk. Okay. So apart from all these pathogens, physical, chemical contaminants, there are food infections, illness because of allergens, because of some food allergens. Sometimes we are allergic to foods. some kind of food we eat. In other countries, maybe when you travel, you may be familiar that in peanuts they put allergic means you don't eat. Gluten, some are allergic to gluten, the protein in your wheat. Some are allergic to that. They will have some kind of discomfort symptoms soon after eating gluten food because of the protein. Some are allergic to the casein in the milk. So as I mentioned, some are allergic to the histamine in the fish. So lots of reasons are there. These kind of allergens are also should be addressed in case of foodborne illness, right? Then, so now what is the reason? We have already, I have already explained you. So one should know how to prevent it. And first thing, better prevent the entry of them into your food. If at all, if it has entered, prevents its multiplication or proliferation. So first thing, avoid them. If you can't avoid them, try it. Once it has entered, if you suspect it, cook it thoroughly. Try to avoid their multiplication by properly maintaining its danger zone, right? The danger zone, as I mentioned, is 4 degree to 60 degree is the danger zone. So avoid storing the food in this 4 degree to 60 degree so that you can avoid, because some of the bacteria can grow till 50 degree also. So if at all, try to store them in fridge or freezer. And how you have to handle, it's a very important concept. It's a very simple concept one should know in food science is this 4 rules for food safety. Clean. Wash your hands thoroughly before you are cooking, starting your cooking preparations. Whoever is going to handle the food, clean your hands. Wash your hands thoroughly. Whatever surfaces, the kitchen surfaces, the boards, whatever you are using, thoroughly clean them. Especially in the hotels, they have to disinfect them, bleach them thoroughly, bleach the surface. Okay, with a bleaching solution with 1 liter in at least 4 or 5 liters, they have to mix it and thoroughly disinfect the whole cooking surface areas. Then separate. What is this separate? See, we are all, each and every house, we have this refrigerators. Whenever we use the refrigerators, we should use our common sense in preserving, like keeping in the shelves. See, when you are keeping some meat, raw meat, always keep them in the bottom of your refrigerator. So that if you are going to keep them on the top of your refrigerator, what will happen? There is every chance of cross-contaminating. They may spill and contaminate your vegetables or fruits. If you just think about it, if you are going to eat them raw. Okay, so separation is very important. How to separate your raw meats from the other foods is very, very important in your refrigerator. Then cooking at the right temperature is very important, and chill. So refrigerator should not be overloaded, and always don't leave your food which is cooked for more than 2 hours outside. And washing hands also, we all know 20 seconds you need to wash your hands thoroughly. And again, whatever gloves you are using, finger cots you are using, that should also be very clean. And if you are going to eat the food from your refrigerator, see to that you reheat only once. Beyond that, again and again, don't store them and eat them. It is not good for you if there is any toxin in that. And again, if you're going to keep some food in the freezer and you want to cook it, don't quickly bring them to the kitchen top. Don't bring them directly. See, because it will take a long time for them to defrost. Don't do that. See, the time it takes to defrost, it is very convenient time for the bacteria also to multiply. So the best practice to thaw your frozen food is keep them in refrigerator the previous day. So bring them to that temperature, then bring them out. Or keep it in running water or microwave them and then cook it. So leaving the food raw one in outside temperature for a longer time is not good. So clean, separate, cook and chill, you have to know in food safety. So as mentioned, the symptoms of food poisoning, diarrhea, nausea, weakness, abdominal cramps are all very common. So all you need to them is if there is any the episodes is more than 8 times in a day, if there is a watery stool, you have to meet the doctor for rehydration. Depending upon the kind of infection, the medication will be given by the doctor. For virus, I don't think what is the medication they can give. There is no anything like that. They can rehydrate you, and if there is any symptom of diarrhea or nausea or vomiting, try to avoid going to your working environment, especially if you are working in restaurants where you are serving food or working related with food. Even to the office environment, if you go, see, whenever you are using the bathrooms and you are back, wash your hands thoroughly, because you may be spreading the infection to the other person who is sharing the bathrooms with you. So then what you can do is, whenever you eat fruits and vegetables, wash them thoroughly, wash your hands thoroughly, and avoid outdated foods, like just don't store foods for a longer period and don't eat them. And these are all very, very important, right? So these are all the symptoms. You can be have an idea about it. And these are all the symptoms very common to bacteria, virus, and parasites. Okay, so then how to diagnose them? See, very rarely we are doing this, right? Unless the symptom, unless we have this discomfort for a longer period in our country, rarely we go get it tested, isn't it? That is, and also the right time of going for the test, when the fever is high, or else it is very difficult to trace them in your cultures, especially in the blood, in the stool. Then we go for microscopic testing. That is also there. How it is being done is a simple agar plate, they will be grown, and sometimes they will examine your stools under microscopes. And now we know all this RT-PCR test and all, isn't it? So sometimes they are very expensive, and all with a simple genetic markers you can detect the viruses. And these are all the simple tests when we are talking about foodborne illness, not on the other perspectives. Okay, so you need to do that, then only you will know what is the infection. Then if there is see, these are all very, very important when there is an outbreak. See, when what do you call something as an outbreak here? When a group or 1 or 2 individuals are getting the same kind of infection after eating a similar kind of food, we call it as an outbreak, isn't it? And whenever there is an outbreak, it is very important. Now we have this thorough knowledge. So what is the reason for this outbreak? You have to define the cases. So that is very, very important. You have to gather the information, and you have to respond to a foodborne illness outbreak. You have to gather the information. You have to segregate which product is really responsible for this kind of foodborne illness. It's very important to trace, but it all depends upon what is that particular organism, and you have to know what food, where you have eaten, whether the group of people got infection after attending a particular marriage, or who has actually cooked the food, what is being cooked. It's very difficult, and unless otherwise we don't give information to the government, it is really very difficult for the government also to gather the information. Then you have to identify the individual, and you have to take an interview of that particular individual. That is how it was like traced in Kerala, the first COVID positive incident, right? So then how you have to respond, as I mentioned, you have to identify, then you have to proceed the investigation, and proper action need to be taken. So this is a very important concept one should know in food safety, and this we call as ASAP, Asset Analysis Critical Control Points. Whoever wants to take up a job as a food manager of star hotels, or this job opportunity is very, very wide. You have to complete a course on ASAP. I have completed this in Rutgers University. you can take up this course. If you have a degree in food in biotechnology, along with that, if you have completed a certification course in ASAP, you can take jobs like food managers in restaurants and food industries. Okay, it's a well paid job, and it is very mandatory. You have to complete this certification course if you want to take up jobs in food industries. Okay, so we call that course as ASAP, Asset Analysis Critical Control Point. So what is this ASAP actually? ASAP is it is all about prevention, right? What you are preventing, you should know what is the hazard to your food. What is the procedure you can take to control that hazard, so that you can prevent the foodborne illness in food processing plant. So wherever there is large scale food processing plants, industries, if you are a certified ASAP holder, you can work there, right? So it is a state of the art approach to consistent safe food production. So ASAP is proactive, and it is a prevention oriented, focusing on preventing and controlling food safety assets. So the assets are physical, chemical, biological. All these three will be identified, and it will be prevented before it is going to be a real problem. So how this ASAP started, you have to know about it. Actually, this ASAP started in late 1960s. This is this ASAP was originally developed for use in the aerospace manufacturing under the name failure mode effect analysis. You know the Pillsbury Atta Company, Pillsbury Company, which we know in the name of Atta. In 1959, in a project for the NASA space program, this ASAP was used to guarantee the food for the US space program. The food should be 100% free of viral and bacterial pathogens. This is how this ASAP started. So by the late 1960s, Pillsbury was using an ASAP system to manufacture consumer goods. And in late 1989, the Food Safety and Inspection announced its intention to implement the ASAP in meat operations, poultry inspection operations. Okay. Food Safety Inspection Services, FSIS. We say as FSIS. So this is all how this ASAP has entered into the food industry. So in ASAP, you can see 7 principles are there. The 7 principles in 14 steps will be incorporated. So as I told you that see, when you're working in a food industry as a food manager, what you have to do is you have to work. You have to first form a group. Then you have to instruct your group to conduct an asset analysis. So asset analysis, the team will be done, right? So first thing is make a team. The asset team will focus on assets, how it can be prevented, how it can be eliminated, how it can be controlled by a proper asset planning. Then the second step, what they will do is they will identify the second step. They will identify the critical control points. See, that is nothing but a procedure where you can control your food safety assets, where the asset can be prevented, eliminated, or even reduced to acceptable level. So the team will be doing that using a critical control point decision tree. With that, they will try to control each and every single asset. Then the third principle in ASAP is establish critical limit. So as I mentioned, the physical, chemical, biological parameters must be controlled, isn't it, at a critical control point. So it includes, it can be the time of exposure of your food, it can be the temperature that food particularly should be cooked, what should be the water in that food, what should be the pH of that food. All these, they should make a regulatory standard. With scientific literatures, they should establish the critical limits. Then they have to monitor. The fourth principle is the entire asset team should monitor the procedures, whether all the CCP and CCLs are being correctly taken into control. So monitoring procedures will describe how the measurements will be taken, who is responsible, how frequently these measurements need to be done during the production. See, somewhere the raw material will be taken. For example, you are collecting food grains. Are you collecting the right food grains from the farm? Then how you are processing, and then how you are distributing till it reaches consumers. It is the responsibility of the asset team to give a safe product to its consumer. Whatever food they prepare, they should know what is the asset for that food. First thing, identify the asset. Then how it control, how you are monitoring, how you are making the corrective actions to remove those assets, right? The fifth thing is establish corrective actions. So you have to follow the procedures. You have to follow. The entire team will identify the steps that will be taken to prevent potentially hazardous foods from entering the food chain. So that is very important. The sixth principle in ASABIL is verification. So those activities other than monitoring that determine the validity of the asset plan, whether the system is operating according to the plan, the team should identify the activities. Like they have to audit the critical control points, records, reviews, shipment reviews, instrument calibration, product testing. Every part of the activity should be verified. That we call as the sixth principle of asset analysis critical control point. The seventh principle is record keeping. So a record keeping will always be used to prove that the food was produced safely only. In between, something would have happened that they can prove it. The record will also include information about asset plan, and the record include information of the asset team, what is the product, its description, the flow diagrams, the asset analysis, what corrective actions they have taken, whether they are maintaining the record, whether they are verified. Every detail will be there, right? So when you take in our country, we have the food safety, isn't it? Food Safety Standard Authority of India is there, which is a regulatory framework where foodborne illness is not a local issue. It is a coherent approach. It is required to tackle the challenges very effectively. We have a Food Safety Standard Act 2006 with rules, and Act 2011, the Food Safety Standard Act and Rules and Regulation 2011, where they monitor and surveillance programs are taken to make your food free from pathogens, toxins, pesticides, any other contaminant, right? So a thorough duty of assessment of potential foodborne risk are being undertaken by the FSSAI. There are lots of food jobs now. They are like in case of FSSAI, lots and lots of placements are coming up. There is a common exam where you have to appear to become a food safety officer. Then, so as I mentioned, in case of foodborne illness, the responsibility starts from the farm till the plate. So where the food is produced, how the food is produced, what is the raw material, how you are processing, how you are carrying them, whether in between anything had happened, and how you are marketing, where you are distributing. Somewhere in Africa, it would have been produced. Somewhere in the part of down Tamil Nadu, somewhere we must be eating that product, isn't it? So you have to be very, very careful not to disturb the food we eat. That should no way enter the food chain. So care should be taken from the farm to the fork. That's very, very important, right? Because now global mobility is more. We are traveling a lot. We are eating wherever we want. So it is very, very important. The food we eat is safe for the individual. It is the right of every individual, isn't it? So as I mentioned, these are all the steps you can take to prevent a food infection. So you can avoid the cross-contamination by keeping your utensils, cutting boards, hands and equipments clean. And keeping the food in the proper danger zone is also very important. And to conclude, it's very important, everyone should have the right to an adequate supply of safe, nutritious food, and encourages like WHO states to implement and keep national and when appropriate regional mechanisms for foodborne disease and surveillance. So we have to cooperate as individuals to the local government, and the local government should cooperate with every individual at the next level. So government should take necessary measures to ensure availability of safe food for all in order to sustain the health and the economic development of the people. So what we need, a continued surveillance for foodborne disease outbreaks is very, very important to reveal trends in the foods, regions, and the pathogens associated, right? So of course, we do have whole genome sequences now so that we can detect foodborne disease outbreaks rapidly. And a knowledge and skill is also very important, okay? And all we as an individual, food safety education is very important to the people so that the foodborne illnesses could be definitely minimized. And to end with a quote, each and every member of the food industry, from the farm to the fork, they must create a culture that food safety and nutrition is paramount. And thank you. I stop here. Dr. Renu?
Yes, ma'am. Yes, ma'am.
Can I continue with the fellowships, or shall I continue with the fellowships, or should I have to have a break now?
No, ma'am. We have a few questions. Alfred will tell you the questions. You can answer them for the students.
Is it there in the chat box? Should I stop sharing?
You
you can
stop sharing, ma'am. You can be visible also.
Yeah, fine.
you can turn on your camera.
One second. I'll do that. Yes.
Yes, ma'am. Thank you for this wonderful lecture and enlightening us on the topic foodborne illness and its conditions in today's world. So we have few questions from our participants, and it's in the chat box. So I'll just ask you, ma'am. Ma'am, can you please let us know the role of common food in outbreak of zoonosis and reverse zoonosis, if there's any? Ma'am, you can unmute your mic, ma'am.
Thank you. Yes. Yeah, see, maybe they may be asking about the Nipah virus and all. See, here what you are talking about, the foodborne illness, is completely about food. See, whenever our food is infected directly, we talk about foodborne illness. Talking about zoonotic is something different. Okay, in case of Nipah virus and all, the Pteropus giganticus, that is I don't want to talk about that. Through bat, it got infected. So the palm, like from the sap, this bats used to disturb the sap collectors from the toddy. Toddy they used to collect, right? They used to disturb that by they feed that, and they got those viruses, and their shedding was like infecting the pigs. So those pigs who were rearing the pigs, they got that infection. That is a different case. So that zoonotic, we need not have to confuse with food-borne illness. Food-borne illness happens three ways: fecal-oral route, that is a sewage contaminated food, by food handlers, and third part is zoonotic, of course. Okay. Then,
so one more question we have, like, is it safer to store freshly purchased uncooked meat in the fridge, or do we need to use refrigerator, freezer instead?
Whatever you want, you can do it. The only thing is where you are keeping is very important. How you are starting your cooking is very important. See, of course, nowadays all are working, and we need to store, and that too in conditions like lockdown, we have to keep it. So as I mentioned earlier, if you are keeping your raw meat, see, don't marinate and keep them outside. Even if you marinate it, keep it refrigerated. Okay, in refrigerator, you keep them in the bottom so that maybe you are using a cooked food also in your refrigerator. They should not mix with each other, because unless we are cooking, we don't know what is there in our meat. Okay, so that should not cross-contaminate your cooked food or whatever food like fresh fruits and vegetables which you are going to eat without cooking. So separate them. Okay. Another thing, if we marinate also, we have the habit of keeping fish marinated, leaving it outside. First thing we will marinate, at the last we will fry it. Our parents used to do it, isn't it? Don't do that. The 2 hours time, more than that, and our temperature is very, very sufficient for them to multiply. Okay. Then again, keeping in frozen condition. I told you, when you are keeping thawing, bringing them, defrosting is very important. When you defrost them, it will take a long time. So you will be keeping on the kitchen top. That is not good. That time is also like the food will be chill, but bacteria are capable of multiplying. Right? The danger zone, I told you, 4 degree to 60 degrees is a danger zone for multiplication. So you do keep it in refrigerator, then bring them. If you want to cook, microwave and cook it. Bring it to the normal temperature, or keep it in running water, cold running water, and then start cooking. Don't keep them outside for longer period of time. We all know the generation time of these bacteria. Then.
And one more question, like we usually tend to kill microorganism present in food by cooking, and like, is there any process like when we are cooking, is there anything like we lose the nutrient contents and like of biomolecules present in the food?
You're talking about what food? Any food? Yes, of course. Safe food is very important. As long as your food are safe, cooking them at the right temperature is very, very important, because you need not know the incubation period of your particular pathogen. So cooking in the right temperature was the reason for not having any illness from our country. Nowadays, all these illnesses are becoming common to us. Nowadays, all these illnesses are also common. Colon cancer, that is different. That is not foodborne illness I'm talking about. So the wrong food eating practice. Okay, so cooking the food at the right temperature is very, very important. Mostly in our style of Indian cooking, I think we have a thorough good practice of cooking. I don't think we have the practice of eating very raw fruit, raw veg, like fruits, nothing like that. We never have this habit of taking with cold milk. We never, like, very rare. The smoothies and all, these are all maybe now people are practicing of taking with unpasteurized milk and all. Normally our cooking practice is good. It is safe. You have to eat that way to avoid this foodborne illness. Okay, so we don't know where we end up with meningitis. So once there is a case of meningitis or a jaundice, we say, so he is a teetotaler, he is not having the practice of consuming alcohol. I don't know why he has got this infection. We don't trace the real root cause behind any such infections. So it is always better to have your food thoroughly cooked, and eating is the best practice. That is different case of taking vitamins, but damaging is all different case. But cooking your food thoroughly is very important, because nowadays the producers are not like that. The products which you are getting are not getting from that kind of environment which you have earlier. Okay? Yes.
Yes, ma'am. And the final question is that if the foodborne illness is not treated properly, can it lead to death?
Sometimes. In case of meningitis, see, death, even a pregnant woman losing her child is a death, right? So abortion is stillbirth is really, it's a thing you have to think about, especially diarrhea. See, again I tell you, the cases are underreported. Data are lacking in our country. Maybe after this kind of pandemics and all, definitely we will have this kind of awareness. So the cases are underreported. See, toxoplasma is common in US countries, means obviously it will be here. But the only aspect I'm telling you again, how you are handling your food is very important. See, one death is also a death. Is where listeria will lead to death. We are not really checking and bringing that report. Okay, so the samples are not being tested and reported. That is the reason. Definitely, somewhere in our country also death is happening because of foodborne illness. Maybe the incubation period, maybe they don't trace. They would have eaten the food. Sometimes it is 24 hours, sometimes it's 6 months before, isn't it? So yes, of course, some are fatal. It all depends upon your immunity. If you are immunocompromised, definitely it is going to affect you. Sometimes it is fatal also.
Yes, ma'am. Thank you for answering these questions. Now I kindly request our lab HOD, Dr. Renuka Vishwanathan, to give the vote of thanks.
Yeah, thank you, Alfred.
Such a long time.
Good evening to everyone. On behalf of the Department of Biotechnology, St. Joseph's College of Engineering, it's an honor and a privilege to propose a vote of thanks on this occasion, BioTalk Series version 2.5 on foodborne illness. First of all, I would like to thank God for the successful sailing of this event, and I'm grateful to our management, St. Joseph's College of Engineering, Chairman Dr. V. Babu Manoharan, Managing Director Ms. Jessipriya ma'am, and Director Mr. Sasi Shekhar for giving us this opportunity to host this event. I wish to thank our Principal, Dr. V.S. Rao, and Dean Dr. M. Parvatavardhani for their constant motivation. My sincere thanks to our HOD, Dr. G. Srikumar, for his unstinted support and guidance. I extend my thanks to my department colleagues for their cooperation. My heartfelt thanks to the Speaker of the Day, Dr. Anbaragi, for immediately accepting my request to deliver the talk and for a very informative, detailed session on foodborne diseases and hazard. My special thanks to the student organizer, Mr. Alfred, for the smooth conduction of the event. My warm thanks to all my students for participating in spite of their university examinations and for their evincing interest in learning. Thank you all. And with a small quote, learning is a treasure that will follow its owner everywhere. Thank you.
Thank you, ma'am. I once again thank all the participants for attending this webinar, and I would like to inform everyone that the next BioTalk series version 2.6 will be held on 26th June. Thank you.
Thank you, ma'am. Thank you very much.